Let's learn about nerve system of human. Organization of the nerve system. Nerve system coordinate and control the activities of animals. The two main subdivisions of the nerve system are the central nerve system, which consists of the brain and spinal cord, and the peripheral nerve system, which indicates all nerve tissues outside the central nerve system. Next, components of the peripheral nerve system include cranial nerves and their branches, spinal nerves and their branches, ganglia and sensory receptors. The peripheral nerve system may be subdivided further to the somatic nerve system. The somatic nerve system consists of neurons that conduct impulses from somatic and special sense receptors to the CNS and motor neurons from the CNS to skeletal muscles. The action of SNS or somatic nerve system is voluntary. Then an autonomic nerve system, ANS. The ANS consists of sensory neurons that convey information from autonomic sensory receptors, motor neurons that conduct nerve impulses from the CNS to smooth muscles, cardiac muscle, and glands. Because its motor responses are not normally under conscious control, the action of the ANS is involuntary. The motor part of the ANS consists of two branches, the sympathetic division and the parasympathetic. With a few exceptions, effectors receive nerves from both divisions, and usually the two divisions have opposing actions. For example, sympathetic neurons increase heart rate and parasympathetic neurons slow it down. So in general, the sympathetic division helps support exercise or emergency actions, so-called fight or flight responses, and the parasympathetic division takes care of rest and digest activities. Then an enteric nerve system is there, ENS. The ENS consists of neurons whose enteric plexus in the gastrointestinal tract that functions somewhat as independently of the ANS and CNS. The ENS monitors sensory changes in the controls operations of the GI tract. So SNS is their somatic and special sensory receptors and neurons. Here we could see organization of the nerve system. Subdivisions of the PNS are the somatic nerve system or SNS, the autonomic nerve system or ANS, and the enteric nerve system ENS. So somatic and special sensory receptors and neurons, CNS, brain and spinal cord connections, then somatic motor neurons, voluntary, along with that sensory muscles, like skeletal muscles. Then autonomic sensory receptors and neurons connect with CNS, brain and spinal cord, then autonomic motor neurons like involuntary, sympathetic and parasympathetic division connections, along with that smooth muscle, cardiac muscle and glands at the end. Later, sensory receptors and neurons in GI tract and enteric plexus. These are the sensory parts of PNS. Then motor part of PNS include enteric motor neurons, involuntary in enteric plexus. Then effectors like smooth muscles, glands and endocrine cells of GI tract. Then histology of nerve tissue. Nerve tissue consists of two types of cells, neurons and neuroglia. Neurons provide most of the unique functions of nerve system, such as sensing, thinking, remembering, controlling muscle activity, and regulating glandular secretions. Neuroglia supports, nourishes, and protect the neurons. Then neurons nerve cells possess electrical excitability, the ability to respond to stimulus and convert it into an action potential. A stimulus is any change in the environment that is strong enough to initiate an action potential. An action potential or nerve impulse in an electrical signal that propagates or travels along the surface of the membrane of a neuron. Nerve impulses travels at speeds ranging from 0.5 to 130 meters per second. Parts of neuron. Most neurons have three parts like a cell body, dendrites and an axon. The cell body, perikaryon, contains a nucleus surrounded by cytoplasm that includes typical cellular organelles such as lysosomes, mitochondria and Golgi complex. The neuronal cell bodies also contain free ribosomes and prominent clusters of rough endoplasmic reticulum termed vessel bodies. A nerve fiber is a general term for any neuronal process or extension that emerges from the cell body of a neuron. Most neurons have two kinds of processes like multiple dendrites and a single axon. Dendrites are the receiving or input portions of a neurons. They usually are short, tapering and highly branched. The single axon of a neuron propagates nerve impulses towards another neuron and muscle fiber or a gland cell. An axon is a long, thin cylindrical projection that often joins the cell body at a cone-shaped elevation called the Axon hillock. An axon contains mitochondria, microtubules, and neurofibrils. Because rough endoplasmic reticulum is not present, protein synthesis does not occur in the axon. The cytoplasm of an axon, called axoplasm, is surrounded by a plasma membrane known as the axolemma. Lemma in the sense sheath or husk. Along the length of an axon, side branches called axon collaterals may branch off, typically at a right angle to the axon. The axon and its collaterals end by dividing into many of the processes called axon terminals, like telodendria. Here you could see a picture which represents basic neuron design, like dendrites are there, cell body region, axon hillock, then myelin sheath, axon, node of Tranvia. You could see those portions. Then the site to communication between two neurons or between a neuron and an effector cell is called a synapse. Then structural diversity neurons. Neurons display great diversity in size and shape. Structurally, neurons are classified according to the number of processes extending from the cell body, like multipolar neurons usually have several dendrites and one axon. Most neurons in the brain are spinal cord are of this type. Then bipolar neurons have one main dendrite and one axon. They are found in the retina of the eye, in the inner ear, and in the olfactory area of the brain. Then unipolar neurons are sensory neurons that begin in the embryo as bipolar neurons. During development, the axon and dendrite fuse into a single process that divides into two branches a short distance from the cell body. Both branches have the characteristic structure and function of an axon. So functionally, neurons are classified as afferent sensory, efferent motor, or interneurons, mixed according to the direction in which they transmit impulses related to the central nervous system. So afferent or sensory neurons carry impulses from peripheral sense receptors to the CNS. They usually have long dendrites and relatively short axons. Then efferent or motor neurons transmit impulses from the CNS to effector organs such as muscles and glands. Efferent neurons usually have short dendrites and long axons. Then interneurons are located entirely within the CNS, in which they form the connecting link between the efferent and efferent neurons. They have short dendrites and may have either a short or long axon. Next, neuroglia. Neuroglia or glia cells make up about half the volume of the CNS. Generally, neuroglia are smaller than neurons and they are 5 to 15 times more numerous. In contrast to neurons, glia do not generate or propagate action potentials and they can multiply and divide in the mature nervous system. In cases of injury or disease, neuroglia multiply to fill the space formerly occupied by neurons. Of the 6 types of neuroglia, 4 is like astrocytes, then oligodendrocytes, microglia, and ependymal cells are found only in the CNS. The remaining 2 cells like Schwann cells and satellite cells are present in the PNS. Then myelination, axons surrounded by a multi-layered lipid and protein covering called the myelin sheath are said to be myelinated. The sheath electrically stimulates and insulates the axon of a neuron and increases the speed of nerve impulses conduction. So axons without such a covering are said to be unmyelinated. There are 2 types of neuroglia produce myelin sheath, like Schwann cells in the PNS and oligodendrocytes in the CNS. Each Schwann cell wraps about 1 millimeter of a single axon's length by spiraling many times around the axon. Eventually, multiple layers of glial plasma membrane surround the axon, with the Schwann cells, cytoplasm, and nucleus forming the outermost layer. The inner portion, consisting of up to 100 layers of Schwann cell membrane, is the myelin sheath. The outer nucleated cytoplasmic layer of the Schwann cell, which encloses the myelin sheath, is the neurolemma, like sheath of Schwann. A neurolemma is found only around axons in the PNS. Gaps in the myelin sheath, called the nodes of Ranvier, appear at intervals along the axon. Each Schwann cell wraps 1 axon segment between 2 nodes. The amount of myelin increases from birth to maturity as it greatly increases the speed of nerve impulse conduction. Then speed of conduction of nerve impulses is affected by three factors like temperature. Higher the temperature, the faster the speed, then axon diameter. Larger the diameter, faster the speed, then myelin sheath present only in vertebrates. Then gray and white matter. White matter is composed primarily of myelinated axons. The whitish color of myelin gives white matter its name. The gray matter of the nerve system contains neuronal cell bodies. It has dendrites and myelinated axons, axon terminals, and neuroglia. This appears grayish rather than white because the cell bodies impart of gray color and there is little or no myelin in these areas. In the spinal cord, the white matter surrounds the inner core of gray matter is shaped like a butterfly or the letter H. In the brain, a thin shell of gray matter covers the surface of the largest portion of the brain, the cerebrum and cerebellum. The spinal cord is located within the vertebral cavity of the vertebral column. Then meninges. The meninges are three connective tissue covering that encircle the spinal cord and brain. The spinal meninges surround the spinal cord and are continuous with the radial meninges which encircle the brain. The outermost meninx is the dura mater that is composed of dense irregular connective tissue. The middle meninx is a vascular covering called arachnoid matter because of its spider web arrangement of delicate collagen fibers and some elastic fibers. Then the innermost meninges is the pia mater, a thin transparent connective tissue layer that adheres to the surface of the spinal cord and brain. Between the arachnoid matter and pia mater is the subarachnoid space which contains cerebrospinal fluid. Then protective coverings of the brain. The radium and radial meninges surround and protect the brain. The radial meninges are continuous with the spinal meninges, have the same basic structure and bear the same names: the outer dura mater, the middle arachnoid matter, and the inner pia mater. However, the radial dura mater has two layers. The spinal dura mater has only one. Now we are going to learn about brain organization. The brain and spinal cord develop from ectoderm arranged in a tubular structure called the neural tube. The anterior part of the neural tube expands and constricts a pair that create three regions called primary brain vesicles like prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, hindbrain. The mesencephalon gives rise to the midbrain and aqueduct of the midbrain. Both the prosencephalon and rhombencephalon subdivide further, forming secondary brain vesicles. The prosencephalon gives rise to the telencephalon and diencephalon, and rhombencephalon develops into the metencephalon and myelencephalon. The telencephalon develops into the cerebrum and lateral ventricles. The diencephalons form the thalamus, hypothalamus, and epithalamus. The mesencephalon becomes the pons, cerebellum, and upper part of the fourth ventricle. Finally, the myelencephalon form the medulla oblongata and lower part of the fourth ventricle. So we could see regions like this, like development of brain when we look at this. Walled cavity, then 3-4 week embryo. Brain development is shown in the first figure. Like prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, that is hindbrain regions are shown. When that embryo matures into 5-week embryo, we could see differences like telencephalon is there, cerebral hemispheres are there, walls, diencephalon, thalamus, hypothalamus, and epithalamus. Mesencephalon region forms the midbrain, metencephalon and myelencephalon is formed, pons, cerebellum, along with medulla regions. Then cerebrospinal fluid. Cerebrospinal fluid or CSF is a clear colorless liquid that protects the brain and spinal cord from chemical and physical injuries. It also carries oxygen, glucose, and other needed chemicals from the blood to neurons and neuroglia. CSF continuously circulates through cavities in the brain and spinal cord and around the brain and spinal cord and supraarachnoid space between the arachnoid matter and pia matter. CSF-filled cavities within the brain are called ventricles. A lateral ventricle is located in each hemisphere of the cerebrum. The third ventricle is a narrow cavity along the midline, superior to the hypothalamus and between the right and left halves of the thalamus. The fourth ventricle lies between the brainstem and the cerebellum. Here we could see portions like cavities of the brain lateral ventricles are there, third ventricle, aqueduct of the midbrain, then upper part of fourth ventricle, then lower part of fourth ventricle. Then CSF contains glucose, proteins, lactic acid, urea, cations like Na+, K+, Ca2+, Mg2+, and anions. It also contains some white blood cells. So, formation of CSF in ventricles. The sites of CSF production are the choroids plexus, networks of capillaries in the walls of the ventricles. The capillaries are covered by ependymal cells that form cerebrospinal fluid from blood plasma by filtration and secretion. Next topic, circulation of CSF. The CSF formed in the choroidus plexus of each lateral ventricle flows into a third ventricle through two narrow over openings. The intraventricular foramina. More CSF is added by the choroidus plexus in the roof of the third ventricle. The fluid then flows through the aqueduct of the midbrain, like cerebral aqueduct, which passes through the midbrain into the fourth ventricle. The choroidus plexus of the fourth ventricle contributes more fluid. CSF enters the supraarachnoid space through three openings in the roof of the fourth ventricle, a median aperture and the paired lateral aperture, one on each side. CSF then circulates in the cerebral canal of the spinal cord and in the supraarachnoid space around the surface of brain and spinal cord. Now let's learn about major parts of the brain. The adult brain consists of three major parts like forebrain, midbrain, and hindbrain. Forebrain consists of olfactory lobes, the cerebrum and diencephalon, and hindbrain consists of cerebellum, medulla oblongata, and pons. So cerebellum, diencephalon, and cerebrum collectively known as brainstem. The brainstem is continuous with the spinal cord and consists of the medulla oblongata, pons, and midbrain. Posterior of the brainstem is the cerebellum. Superior to the brainstem is the diencephalons, which consist of thalamus, hypothalamus, and epithalamus. Supported on the diencephalon and brainstem is the cerebrum, the largest part of the brain. So, forebrain consists of olfactory lobes. Olfactory lobes are paired short, club-shaped structures present at the anterior part of the brain. Cavity in olfactory lobe is called rhinocele. A pair of olfactory nerve arises from the olfactory lobes. They are not well developed in human brains. Then cerebrum. The cerebrum is the largest part of the brain and controls voluntary actions, speech, thought, and memory. The cerebrum is divided into two halves known as the right and left hemispheres. A mass of fibers called corpus callosum links the hemispheres. The right hemisphere controls voluntary limb movements on the left side of the body, and the left hemisphere controls voluntary limb movements on the right side of the body. Each hemisphere is divided into four lobes or areas which are interconnected. The frontal lobes are located in the front of the brain and are responsible for voluntary movement, and via their connections with other lobes participate in the execution of sequential tasks, speech output, organizational skills, and certain aspects of behavior, mood, and memory. Then the parietal lobes are located behind the frontal lobes and in front of the occipital lobes. They process sensory information such as temperature, pain, taste, and touch. In addition, the processing includes information about numbers, attentiveness to the position of one's body parts, the space around one's body, and one's relationships to this space. Then the temporal lobes are located on each side of the brain. They process memory and auditory, like hearing information, and speech and language functions. The occipital lobes are located at the back of the brain. They receive and process visual information. The surface of each cerebral hemisphere shows many convolutions called gyri, located, like separated by depressions called sulci. The cerebrum consists of two surfaces like cerebral cortex and cerebral medulla. Cerebral cortex is the outer layer of the cerebrum. It is made of grey matter and contains many layers of nerve cells. The inner surface is called cerebral medulla, which is made up of bundles of myelinated axons, the white matter. Then diencephalon, it contains the epithalamus, thalamus, and hypothalamus. Epithalamus is non-nervous part which is fused with pia mater to form anterior choroid plexus. Then thalamus directs sensory impulses from the lower parts of the brain and spinal cord to appropriate parts of the cerebrum. Limited sensory awareness of pain, temperature, touch, and pressure is provided by the thalamus. And hypothalamus presents just beneath the thalamus and forms the floor and the part of the lateral walls of third ventricle. It contains reflex centers linked to the autonomic system. They include the thermoregulation centers for temperature regulation, appetite and satiety center to control the desire for food, then controls sleeping patterns and wakefulness. Along with that, hypothalamus secretes neurohormones which control the secretions of anterior pituitary hormones. It synthesizes the posterior pituitary hormones and controls their release into the blood. The next region, midbrain. Midbrain is very small constricted portion of brain that is covered by the cerebrum. It is represented by a pair of longitudinal bands of nerve tissue, crura cerebri on ventral side, dorsally a pair of small swellings called corpora bigemina on either side, and four lobes, optic lobe, are collectively called corpora quadrigemina, in which superior colliculi are related to optical activity, while the two smaller posterior colliculi to the auditory. These two are collectively known as tectum. Then hindbrain region, cerebellum. Cerebellum is the second largest part of brain and is located at the back of the skull. It consists of a pair of cerebellar hemispheres and a small median vermis. The grey matter of cerebellum is called cerebellar cortex, while the white matter inside formed a branched T-like structure called arbor vitae. Then I'm not sure about the pronunciation. Then function of cerebellum. It helps control body posture, maintenance of muscle tone, coordinate voluntary muscular activities and equilibrium of body. Then pons. The pons lies directly superior to the medulla and anterior to the cerebellum. Pons serves as a bridge between the midbrain and the medulla oblongata. The pons also contains nuclei and fibers of nerve that serve eye muscle control, facial muscle strength and other functions. Then medulla oblongata. The medulla oblongata is the lowest part of brainstem and is interconnected with the cervical spinal cord. The medulla oblongata also helps control involuntary actions including vital processes such as heart rate, blood pressure and respiration, and it carries the corticospinal, that is motor function tract, toward the spinal cord. Here we could see major parts of the human brain like cerebrum, corpus callosum, midbrain region, cerebellum, pons region, medulla oblongata. Next we are going to learn about spinal cord. The spinal cord is an extension of the brain and is surrounded by the vertebral bodies that form the spinal column. The spinal cord is connected to a section of the brain called the brainstem. The cerebral structures of the spinal cord are made up of grey matter, nerve cell bodies, and the external or surrounding tissues are made up of white matter. Spinal cord conducts impulses to and from the brain and controls most of the reflex activities and provides a means of communication between spinal nerves and the brains. Then peripheral nerve system, PNS, is there. The nerves arising from the central nerve system constitute the peripheral nerve system. Peripheral nerve system consists of somatic nerve system and autonomic nerve system. Somatic nerve system, voluntary nerve system, it is constituted by the nerves which arise from the brain and spinal cord. They are respectively called cranial and spinal nerves. Cranial nerves have 12 pairs in man. Types, origins and nature as well as functions are given below in this table. Whereas there are 31 pairs of spinal nerves. All are mixed sensory and motor, which are formed by the union of dorsal and ventral roots shortly after they leave the spinal cord. Like olfactory, origin olfactory lobe or bulb, nature sensory, smell, then optic lobe on midbrain, nature sensory, function sight, like retina of eye, like that, oculomotor, trochlea, then trigeminal divided into three, like ophthalmic, maxillary, mandibular, like ventral surface of pons varolii, nature mixed, movement of tongue, jaw muscles for chewing and like that, facial, auditory, glossopharyngeal, vagus. Then spinal accessory nerves, hypoglossal. Vagus is the longest and trochlear is the smallest cranial nerve. Then autonomic nerve system. Autonomic nerve system regulates the activities of involuntary smooth muscles, cardiac muscles and glands without consulting the will. The system is entirely motor, having efferent fibers. The system consists of preganglionic fibers from CNS, ganglia and postganglionic fibers. It is also called visceral nerve system because the autonomic nerve system intervenes the viscera. It consists of two antagonistic components like sympathetic and parasympathetic. Now let's learn about the effect of sympathetic and parasympathetic divisions of the autonomic nerve system. Like target organ, digestive system, parasympathetic effects increase smooth muscle mobility, peristalsis and amount of secretion by digestive system, glands, relaxes sphincters. Sympathetic effects decreases activity of digestive system and constricts digestive system sphincters, for example anal sphincter. Then liver, no effect, causes glucose to be released to blood, lungs, constricts bronchioles, dilates bronchioles. Urinary bladder or urethra, relaxes sphincter, constricts sphincter. Kidneys, no effect, decreases urine output. Then heart, decreased rate shows and studies an increased rate and force of heartbeat in case of sympathetic nervous system. Then blood vessels, no effect on most blood vessels but constricts blood vessel and viscera and skin. Dilates those in skeletal muscle and heart. Please read this from the screen. Pause the video if you need to read this carefully. So that's all about this topic of central nervous system and peripheral nervous system. We just had a quick recap about all this topic. If you paid attention, you learned about human brain and central nervous system, peripheral nervous system, some important points about cerebrospinal fluid, cerebral ventricles, and some points related to the cerebrospinal fluid production. So, thank you for listening.